here you are, welcomed softly by Dreamtime Science in the silence of a starlit evening. Picture a quiet library, tall windows revealing the velvety night sky, scattered with shimmering lights far, far away. It's a place where the mind may wander freely, where every word floats like a gentle breeze across your thoughts. If you feel at home in these stories, a quiet like or a subscription would help light our way, and do share a note in the comments about the peaceful place you're listening from. Now, take a moment to find perfect comfort. Adjust your position, perhaps turn over on your side, and breathe in the calm that night brings. We are ready to begin our gentle exploration now, together, letting every whisper of science bring you closer to rest and wonder under this soft, endless sky. In tonight's ethereal voyage of Dreamtime Science, we shall softly unfurl the mystery of our mind's imagery. Can thoughts be recorded like a video? As we glide gently into the realm of cerebral curiosities, let's surrender to the wonder of what lies within our skull's silent cinema. So, my dear dreamers, let's tiptoe into this mystical labyrinth of thought, always maintaining a pillow-soft distance from the harsh edges of reality. Before you drift away, a gentle tap of the like button or a soft whisper of subscription helps keep our nightlight glowing. Where are you joining us from tonight? Leave a dreamy note below. Take this moment to settle in, wrap yourself in comfort, release the day's weight from your shoulders, and let your breath become as gentle as starlight. Thoughts are a fascinating inner experience, a continuous river flowing quietly beneath the surface of our awareness. Although we cannot hold or touch a thought like a physical object, they shape our lives profoundly. They form the foundation of our mental life, the things we think about, the judgments we make, the memories we recall, and the creative insights that suddenly appear. To understand our thoughts is to embark on an exploration deep within ourselves, tracing how ideas and feelings arise, move, and sometimes fade away. At its simplest, a thought can be described as a mental event, a kind of activity happening inside the brain. While externally invisible and intangible, thoughts are fundamentally based on neurophysiology, the complex way billions of nerve cells or neurons interact within our brain. Neurons communicate with each other through tiny electrical impulses called action potentials, which move rapidly along their long, slender projections called axons. When a neuron fires, it releases chemical messengers known as neurotransmitters into small gaps called synapses, passing signals to other neurons. It is this dance of electrical and chemical signals that produces the rich inner life we call thought. But thinking is not mere random firing. The brain is an extraordinarily organized organ, divided into specialized regions that work together seamlessly. Areas like the prefrontal cortex, located right behind your forehead, are especially important for what scientists call executive functions. These include planning, reasoning, decision-making, and the formation of complex thoughts. Deep inside the brain, structures like the hippocampus help in creating and recalling memories, feeding information into the swirling currents of thought. Even parts of the brainstem play a role, regulating alertness and enabling the conditions necessary for conscious thinking. To unpack how a thought forms, imagine that you see a blooming flower. Your eyes capture photons of light reflecting from the petals. This visual information travels through the optic nerve to the visual cortex at the back of your brain. Here, the image is processed and identified as a flower with its vibrant colors and delicate shape. This sensory data is then relayed to regions involved in emotion and memory, linking what you see with past experiences, perhaps a pleasant memory of picking wildflowers on a sunny day. Suddenly, a mental image and feeling arise. A thought about the flower's beauty and your joyful memory. Thoughts are not just pictures or images. They can take many forms. Sometimes they appear as words, our internal voice narrating the world. Other times they are abstract ideas or emotional sensations, an anxious feeling about tomorrow, an excited spark of curiosity, a warm sense of gratitude. The brain can combine these elements in remarkable ways to generate complex thoughts, such as pondering a philosophical question, solving a math problem, or composing a piece of music. This variety shows that thought is deeply multifaceted, involving sensations, language, emotions, and memories all intertwined. Memory plays a crucial role in thought formation. Our brains have different types of memory: sensory memory, short-term or working memory, and long-term memory. Sensory memory holds impressions from our senses for brief moments, allowing the brain to select what is important. Working memory is like a mental scratch pad where information is actively held and manipulated for a short duration, often just seconds to minutes. Finally, long-term memory stores accumulated knowledge, skills, and experiences for days, years, or even a lifetime. When the mind thinks about something, it often calls upon long-term memories as reference points. These memories act as building blocks for new ideas. For example, when you are trying to solve a puzzle, your brain retrieves related concepts and experiences stored in long-term memory, assembles them in working memory, and plays with them until a solution emerges. This process, sometimes called executive function, requires the prefrontal cortex to direct attention, juggle information, and inhibit distractions. Working memory keeps the thought stream coherent and goal-oriented, yet thoughts are not isolated moments. Rather, they flow like a gentle stream, or sometimes a rapid river, connecting one idea to the next, smoothly or abruptly. This continuity is often called the stream of consciousness. It is what gives our mental life a sense of unity, allowing us to plan, reflect, or daydream. Neuroscience suggests this continuity arises because neurons in different brain regions stay dynamically coupled, maintaining communication over time. Brain oscillations, rhythmic waves of neural activity recorded by EEG, also help regulate how thoughts unfold, coordinating groups of neurons to fire in synchrony. One might ask, where do thoughts arise? Are they located in some specific corner of the brain or somehow spread everywhere? The answer is subtle. Thoughts depend on a network of brain regions working in concert, a system neuroscientists call the default mode network, DMN. The DMN becomes active during rest or introspection, when we are not responding to the external environment but reflecting inwardly. Key hubs include the medial prefrontal cortex, the posterior cingulate cortex near the back of the brain, and the angular gyrus. Activity within the DMN correlates with self-referential thoughts, recalling memories, imagining the future, or considering the perspectives of others. Another interesting question relates to the nature of thoughts. Are they discrete packets of information or continuous flows? Research shows both perspectives hold truth in different contexts. Sometimes thoughts arise as distinct ideas, like lighting up briefly in the mind, an answer to a question or a sudden realization. Other times, thoughts unfold gradually and continuously, as when daydreaming or during deep reflection. Brain imaging studies suggest that neural activity can flicker between focused bursts and more diffuse patterns depending on the task, mood, and attention. The chemical environment of the brain also shapes thought. Neurotransmitters like dopamine, serotonin, and norepinephrine influence how thoughts form and feel. Dopamine, often called the reward chemical, is crucial in learning, motivation, and creativity. When dopamine systems activate, thoughts can become more fluid, imaginative, or goal-directed. Serotonin plays a role in mood regulation, influencing whether our thoughts lean toward optimism or worry. Imbalances in these neurotransmitters can contribute to conditions like depression or anxiety, where thought patterns become repetitive or intrusive, showing how deeply emotions are linked to the nature of thinking. It may surprise some to learn that even during sleep, the brain continues its thinking work. Dreams are a mysterious form of thought that combine memories, emotions, and sensations in unique, often illogical ways. During rapid eye movement, REM sleep, the brain activates much of the neural machinery involved in thinking, but with important differences. Certain inhibitory regions tone down, allowing ideas and images to flow freely without the strict logic of waking thought. This helps with emotional processing and memory consolidation, underlining how thinking is a complex process, not limited to conscious awareness. We might also consider the evolutionary roots of thought. Thought allows humans to solve problems, plan for the future, understand others' intentions, and create cultures. Some scientists see human thought as an advanced form of cognition that builds on simpler processes found in other animals. For instance, many animals show signs of memory, problem-solving, and even rudimentary self-awareness. However, the rich symbolic and abstract thinking typical of humans seems to depend heavily on our highly developed prefrontal cortex and language abilities. Language is indeed a remarkable partner to thought. While thought can exist without language, as in pure sensory emotions or abstract feelings, language provides a scaffold to clarify, manipulate, and communicate our thoughts. Words allow the mind to categorize experiences, reason analytically, and share ideas with others. In many ways, internal speech is a cognitive tool, helping the brain rehearse plans, solve problems, and reflect on itself. When we talk to ourselves in our heads, we engage in this internal dialogue that shapes the flow and structure of thought. Yet thought is not simply a closed loop inside the brain. It is intimately connected with the body and environment. Sensory input provides raw material for thinking. Movements and speaking allow us to test and express thoughts. Social interactions influence and refine our mental models. This view, often called embodied cognition, suggests that the brain does not work alone, but in constant dialogue with the body and surroundings. This perspective helps explain why changing posture or even facial expressions can influence our mood and thought patterns. Our thoughts have a curious quality. They appear real and immediate, but are also ephemeral. They come and go, sometimes with great force, other times gently fading away. This transient nature shows that thoughts are more like processes than things. They are patterns of activity, ever-shifting and dynamic, not static entities stored somewhere. This fluidity allows the mind to adapt, learn, and imagine new possibilities, but it also means that mental health depends on delicate balance and regulation. Scientists use many methods to study thoughts. Functional magnetic resonance imaging, fMRI, for example, measures changes in blood flow to different brain regions, showing which areas are active during various mental tasks. Electroencephalography, EEG, records electrical waves across the scalp, revealing temporal dynamics of brain activity underlying thought processes. Emerging techniques like magnetoencephalography, MAG, provide even finer details. Combining these insights with psychological experiments, researchers gain clues about how thoughts emerge, what shapes them, and how they relate to consciousness. Consciousness itself is deeply intertwined with thought, though the relationship remains one of the greatest mysteries. Some theories propose that conscious awareness arises when certain patterns of neural activity reach a threshold of integration and coherence. Thoughts that enter conscious awareness are the ones we can reflect on, describe, and choose to act upon. But much thinking happens below the surface, in subconscious or preconscious states, influencing behavior without crossing into full awareness. This distinction helps explain why sometimes insight feels like a sudden illumination and other times it develops gradually over time. Our understanding of thought is still evolving. New discoveries continue to reveal the brain's incredible complexity and plasticity, the ability to change with experience. Learning shapes neural connections, strengthening some pathways and pruning others. Meditation and mindfulness practices demonstrate how attention and awareness can be trained to influence patterns of thought, reducing rumination and improving well-being. Artificial intelligence research models aspects of human thinking using algorithms that simulate neural networks, though so far they lack the consciousness and subjective richness of our own inner life. To draw this exploration toward a gentle pause, consider that each thought is like a fleeting spark illuminating the vast landscape of your mind. These sparks come from countless microscopic interactions, electrical rhythms, chemical shifts, and neural conversations occurring in synchrony. They carry the weight of your past experiences and the promise of future possibilities. Though invisible, they shape how you perceive, feel, decide, and create. Understanding their nature deepens our appreciation for the remarkable organ within our skulls, where the mysteries of self, perception, and knowledge unfold silently in endless wonder. And so, when your thoughts drift tonight, sometimes clear, sometimes foggy, sometimes wandering down unexpected paths, remember, they are part of a grand symphony playing out in your brain. Each thought is a note in the music of your mind, a moment of conscious awareness born from a billion tiny sparks working together. Sleep gently, carrying these shimmering moments in the quiet sanctuary of your mind, ready to awaken to new daydreams and discoveries with the dawn. There is something truly remarkable about the human brain. Nestled safely within the protective confines of the skull, it quietly orchestrates every sensation, thought, and motion we experience. In many ways, the brain can be thought of as a biological hard drive, a magnificent storage system that not only holds vast amounts of information, but also retrieves and processes it whenever we need to understand our experiences, make decisions, or simply remember a fond memory from long ago. To appreciate this analogy fully, it helps to consider what a hard drive does in a computer. A hard drive stores data magnetically or electronically, holding on to bits of information, whether the machine is powered on or off. When a user wants to access a file, the hard drive retrieves those bits, allowing the computer to display an image, play a song, or open a document. In much the same way, the brain stores memories and then reaches back into that storehouse to bring them to our conscious awareness, whether it's a familiar face, a new skill, or a fleeting thought. But the brain's method of storage is far more complex and fascinating than any mechanical hard drive. The brain doesn't store memories as isolated files in neat folders. Instead, its storage system is distributed across a vast network of neurons, specialized cells designed to transmit and process information. Each neuron can connect to thousands of others via tiny junctions called synapses. These connections form intricate patterns that represent specific pieces of information, whether a phrase in a conversation, the smell of rain, or the feeling of happiness. When we talk about storing memories, it's helpful to divide them into several types. The first and most well-known kind is explicit memory, the kind we consciously recall. This includes episodic memories, which are memories of specific events, your last birthday party, a childhood trip to the beach, and semantic memories, which are facts and general knowledge, such as knowing the capital of France, or how to solve a math problem. Then there are implicit memories, a subtler form stored without conscious effort. These memories govern skills and habits like riding a bike or typing on a keyboard, and even emotional responses. The structure of the brain plays an essential role in these processes, with certain regions specializing in different types of memory functions. The hippocampus, for instance, is deeply involved in the formation of episodic and semantic memories. Situated within the temporal lobe, this small seahorse-shaped area acts like a pivotal hub, encoding new experiences and helping to consolidate them into long-term memories. It works by linking different sensory inputs, what you see, hear, and feel, into a cohesive, retrievable memory. The process of consolidation is a key step in storing memories long-term. Initially, new experiences are fragile and vulnerable to disruption. It might be easy to forget a person's name shortly after being introduced. But over time, repeated activation of the neural connections associated with that memory strengthens the pathways, making the memory more stable and accessible. Sleep plays a crucial role here, acting as a time when the brain rehearses and strengthens these connections. Beyond the hippocampus, the cerebral cortex holds vast stores of semantic knowledge and is where many long-term memories reside. Different parts of the cortex are specialized to hold different kinds of information. For example, visual memories involve the occipital lobe at the back of the head, while auditory memories tap into regions of the temporal lobe. This distributed storage means that a complete memory might simultaneously activate multiple areas of the brain. When retrieving memories, the brain doesn't simply pull out a perfect recording like a video player. Instead, it reconstructs the memory from its neural traces, weaving bits of sensory input, emotions, and associations into a coherent whole. This is why memories can sometimes feel vivid and accurate, but at other times become fuzzy or altered. The very act of remembering can subtly change the memory itself as new experiences blend with old. Now, let's delve a little deeper into the cellular level where the actual storage happens. Each neuron communicates primarily through synapses, where chemical messengers called neurotransmitters shuttle signals from one cell to the next. When we learn something new, or when a memory is formed, synaptic connections strengthen in a process known as synaptic plasticity. Essentially, this is the brain's way of fine-tuning its network, reinforcing the pathways that represent important information. One of the most famous mechanisms of this plasticity is long-term potentiation, or LTP. It's a bit like turning up the volume on a particular synaptic connection, making it easier for neurons to fire together in the future. This concept is often summed up in the phrase, "Neurons that fire together wire together." Imagine repeatedly practicing a musical scale on the piano. The more you repeat it, the stronger the connections become among neurons that represent the finger movements, sounds, and rhythm, embedding this ability into your memory. It's also fascinating how the brain manages to store such immense amounts of information despite its relatively finite size. The secret lies in the combinatorial power of its connections. With around 86 billion neurons intertwined by perhaps 100 trillion synapses, the potential patterns of activity are almost limitless. Each unique pattern can encode a different thought, memory, or concept, allowing the brain to store a tremendous diversity of information within a compact space. While neurons and synapses are the core components, other types of brain cells, like glial cells, also support the storage and retrieval of memories, although their exact roles are still being explored. They help maintain the environment around neurons, provide nutrients, and even participate in modulating synaptic activity. One of the most poetic aspects of the brain's biological hard drive is its ongoing adaptability. Unlike a mechanical hard drive that might fail or degrade over time, the brain has a remarkable capacity to reorganize itself, especially in early life, but continuing throughout adulthood. This neuroplasticity means that if one pathway is damaged, others may sometimes compensate. For memory, it means that learning is a dynamic, continual process. The brain is always updating, remodeling, and refining its storage system, but sometimes the system falters. Memory loss, whether temporary like in déjà vu experiences or more serious as in diseases like Alzheimer's, reveals how delicate and complex the storage and retrieval mechanisms are. Alzheimer's disease, for example, is characterized by the accumulation of abnormal protein deposits that disrupt synaptic function, particularly in the hippocampus and cortex, leading to the inability to form new memories and retrieve old ones. Scientists study memory not only by observing patients with such conditions, but also through experiments that measure brain activity using tools like functional MRI or EEG. These methods reveal patterns of neural activation that correlate with memory tasks, helping us unravel the architecture of memory networks. Interestingly, memories are often linked with emotions, given the close relationship between the hippocampus and the amygdala, the brain's center for emotional processing. This association helps explain why emotional events, whether joyful or traumatic, tend to be remembered more vividly than mundane ones. The emotional tag makes those memories more salient, increasing the likelihood they will be recalled vividly. Memory is also interwoven with identity. Our sense of self relies heavily on continuity and the ability to recall past experiences. This is why disruptions in memory can profoundly impact personality and consciousness. The brain's biological hard drive does more than store data. It holds the narrative of our lives. The gradual unfolding of memory storage and retrieval is fascinatingly slow and meticulous. When a new event occurs, sensory information floods into the brain through the peripheral nervous system and is processed in primary sensory areas. Then, the hippocampus helps bind this information together, marking it for long-term storage, a step called encoding. Over hours to days, this encoded information is consolidated and distributed across the cortex for stable storage. Retrieval then invokes a process of pattern completion, where a partial cue, a familiar scent, a word, or a place, triggers the reactivation of the entire memory trace. This reactivation allows the memory to enter conscious awareness. Unlike the mechanical hard drive, which is static, this biological system is akin to a living library where books are read, highlighted, rewritten, and sometimes misplaced. Beyond memories of facts and events, the brain's hard drive stores endless other information critical to life. It holds learned skills, procedural memories that allow us to perform complex, coordinated actions without conscious thought. It keeps track of language rules, social norms, and even unconscious biases shaped by experience. In this way, the brain is both a database and a dynamic processor, continually weaving past knowledge into present function. At night, during sleep, the brain actively consolidates memories while new sensory input pauses. During deep sleep and rapid eye movement, REM sleep, neurons replay recent experiences, strengthening the synaptic connections made earlier in the day. This replay is thought to help transfer memories from the temporary storage of the hippocampus to the more permanent holdings in the cortex, much like a computer syncing data to its hard drive. In subtle terms, the brain's biological hard drive shines as a testament to nature's ingenuity. It overcomes physical limitations with elegant biological solutions, using networks of adaptable cells, chemical signals, and electrical impulses, all working harmoniously. It holds the blueprint of our existence and the seeds of our future selves. Understanding the brain's role in storing and retrieving thoughts and memories invites a deep appreciation of our own minds. It opens up a space to marvel at the intricate dance of neurons, the plasticity that allows lifelong learning, and the fragile yet persistent nature of our mental archive. So the next time you recall a memory, a lullaby your mother sang, a breathtaking sunset, a lesson learned, you can reflect on how countless neurons and synapses have aligned and fired in your own biological hard drive, making that memory alive again. Thus, the brain serves as our life's ever-accessible archive, quietly powerful, yet exquisitely delicate. It ensures that our past experiences inform our present choices, that knowledge guides our actions, and that the essence of who we are persists across time. And as you drift towards sleep, let this humbling and beautiful system rest alongside you, ready to weave new memories from the dreams that await. It is a quiet marvel to consider how scientists have devised methods to peer inside our brains without ever making a single incision. The art and science of neuroimaging have transformed our understanding of the human mind, the very organ that defines much of our experience. Tonight, we will take a gentle journey through this fascinating world, exploring the tools and techniques that illuminate the brain's hidden landscapes, revealing not only structure, but function and, occasionally, the whispers of thoughts themselves. At first glance, the brain may seem like a dense, enigmatic mass of grey and white matter, but it is much more than that. Every millimeter cubed contains billions of neurons, sending signals in a dance of electrical and chemical activity that produces everything from a fluttering heartbeat to the sensation of joy. Unlocking this complexity has been a challenge stretching back centuries. Historically, thinkers relied on injuries and post-mortem dissections to infer brain functions. While these approaches laid important foundations, the advent of neuroimaging ushered in a new era of non-invasive exploration, enabling us to see the living brain at work. Beginning with the simplest structural brain imaging techniques, allow us to observe the anatomy of the brain in remarkable detail. One of the earliest imaging methods still widely used today is the CT scan, short for computed tomography. The CT scan uses X-rays taken from multiple angles to create a composite cross-sectional image, somewhat like slicing a loaf of bread and looking at each slice carefully. This method excels in revealing gross structural changes such as tumors, hemorrhages, or bone fractures of the skull. However, CT scans cannot show the nuances of brain function or the subtle differences in soft tissue, contrast, as well as more advanced methods. For a clearer picture of soft tissues, magnetic resonance imaging, or MRI, is the gold standard. It uses powerful magnets and radio waves to stimulate hydrogen atoms in our body, primarily in water molecules, to emit signals that sensors then detect. Through sophisticated mathematical reconstruction, these signals transform into detailed, three-dimensional images of the brain's structure. One might appreciate how MRI is particularly useful for examining grey matter, the locations of neuronal cell bodies, and white matter, the nerve fibers connecting different regions. MRI images provide exquisite detail that can reveal conditions like multiple sclerosis lesions, stroke damage, or abnormalities in brain development. But seeing the brain's shape and structure is only part of the story. For centuries, researchers yearned to understand function, to know what parts of the brain light up when we speak, feel emotions, or solve puzzles. This desire propelled the development of functional neuroimaging, a suite of technologies that increasingly allow us to observe brain activity in real time. One of the pioneering techniques in this field is the electroencephalogram, or EEG, which measures the brain's electrical activity. Electrodes placed on the scalp detect the tiny electrical fluctuations produced when neurons fire, It turns out that these signals, although incredibly faint, follow characteristic patterns depending on whether a person is awake, asleep, concentrating, or relaxed. EEG has been invaluable in diagnosing conditions like epilepsy, since seizures produce distinctive electrical bursts. Additionally, it can reveal the general state of cortical activity during sleep stages or anesthesia. However, EEG, like all methods, has its limitations. Because electrodes sit on the scalp, the exact origin of a signal within the brain is difficult to localize precisely. That is, it tells us when activity happens, but lacks fine spatial resolution about where, deep inside the brain, it comes from. Also, it primarily records the summed electrical activity near the brain's surface, making it less useful for seeing deep brain structures. To overcome such spatial limitations, we turn to other functional imaging methods rooted in measuring blood flow and metabolism. The central idea here exploits what is known as neurovascular coupling. When a region of the brain becomes more active, it consumes more oxygen and nutrients, prompting an increase in local blood flow. This physiological response can be detected and translated into images reflecting brain activity. The workhorse of modern brain activity imaging is the functional MRI, or fMRI. Unlike the structural MRI we spoke of earlier, fMRI captures changes in blood oxygenation levels, termed as the bold signal, blood oxygen level dependent contrast. When neurons engage in a task, they demand more oxygen, prompting vessels to deliver more oxygenated blood than needed, causing a relative increase in oxygenated hemoglobin. Because oxygenated and deoxygenated hemoglobins have different magnetic properties, the MRI scanner can detect these subtle differences over time. Over the past few decades, fMRI has revolutionized cognitive neuroscience by enabling researchers to map brain regions involved in perception, language, motor control, emotion, and many other functions without any invasiveness. For example, asking a person to imagine moving their hand allows scientists to observe activity in the motor cortex, while listening to speech activates regions in the temporal lobe. Still, there are important limitations to fMRI to recognize. First, there is a delay. Neuronal electrical events happen in milliseconds, but the blood flow response lags behind by several seconds. This means the temporal resolution of fMRI is slower than neural events themselves. Furthermore, the bold signal is an indirect measure. It reflects vascular changes related to neuronal activity rather than direct electrical signals. Thus, interpreting activity requires care, considering the complex interactions among neurons, blood vessels, and glial cells supporting the brain. Another ingenious technique that measures brain metabolism differently is positron emission tomography, abbreviated as PET. This method involves injecting a small amount of a radioactive tracer, typically a molecule like fluorodeoxyglucose, FDG, which mimics glucose the brain's primary fuel. Active regions use more glucose, causing the tracer to accumulate there and emit positrons detected by specialized cameras. PET provides unique insights into metabolic processes, neurotransmitter systems, and even the accumulation of proteins associated with diseases like Alzheimer's. Although PET scans reveal functional and molecular information unobtainable by other methods, their use is constrained by the need for radioactive substances, exposure to which must be minimized, and by lower spatial resolution compared to MRI. They are often employed clinically or in research studies where understanding metabolism or molecular pathways is crucial. Alongside these established methods, newer technologies continue to emerge, promising to expand our capabilities. Near-infrared spectroscopy, or NRS, uses light to measure blood oxygenation changes in the cortex, much like fMRI, but with portable devices allowing more flexible measurement outside of scanners. Magnetoencephalography, (MEG), detects magnetic fields generated by neural activity, complementing EEG with better spatial localization, yet requiring heavily shielded rooms and sensitive equipment. Let us also contemplate the incredible contributions made by advances in computational neuroimaging. Raw data from EEG, MRI, or PT are complex and noisy. Sophisticated software and algorithms help reconstruct images, analyze connectivity between brain regions, and extract meaningful patterns of activity. Machine learning techniques now increasingly allow scientists to predict patterns of cognition or behavior from neuroimaging data, a frontier that sparks both excitement and ethical considerations. Yet despite these remarkable achievements, neuroimaging is not without profound challenges. One enduring difficulty is separating cause and correlation. When a brain region shows increased activity during a task, does it play a causal role, or is it merely along for the ride? It takes careful experimental design and sometimes combining multiple techniques to approach such questions. Another limitation is the immense complexity and variability of human brains. No two brains are exactly alike. Anatomy and function vary with age, genes, experience, and environment. Interpreting imaging results requires understanding this diversity and avoiding overgeneralization. Furthermore, the brain is a dynamic organ, continually adapting and rewiring itself, so static snapshots risk missing rich longitudinal changes. In clinical practice, neuroimaging has become indispensable in diagnosis, surgical planning, and monitoring treatment outcomes for neurological disorders. It aids in understanding stroke, tumors, epilepsy, neurodegenerative diseases, traumatic brain injury, and psychiatric illnesses. However, clinical scans usually provide macro-level information. Embracing functional and molecular imaging techniques can offer deeper personalized insights, albeit with increased complexity. Looking forward, the potential of neuroimaging teems with promise. ultra-high field MRI scanners operating at magnetic strengths many times stronger than typical clinical machines continue to improve resolution and contrast. Developments in simultaneous multimodal imaging allow EEG and fMRI data to be collected together, integrating temporal precision with spatial detail. Emerging technologies harness techniques like optogenetics and calcium imaging in animal models, enabling direct observation and manipulation of neuronal activity at unprecedented resolution, paving pathways toward translational research. Moreover, ongoing efforts strive to build detailed brain atlases, mapping not only anatomical landmarks but also molecular markers and connectivity patterns. Such comprehensive maps serve as invaluable references for understanding normal brain function and its disruptions. However, with these advances arise questions about privacy and the interpretation of brain data. If we unravel brain activity patterns associated with thoughts or intentions, how do we ethically use this knowledge? Responsible stewardship of neuroimaging data demands thoughtful dialogue and regulation to safeguard individual rights and dignity. In reflecting on these intricate technologies, each a stroke of ingenious creativity married with physics, biology, and engineering, we appreciate how far we have come in illuminating the brain's mysteries. From measuring electrical pulses with scalp electrodes to witnessing oxygen-rich blood coursing through neuronal circuits, each method provides a different lens through which to view the symphony of cognition unfolding within us. Before closing, it is rewarding to remind ourselves that neuroimaging is not about creating pictures for picture's sake. The ultimate goal is to deepen our understanding of the mind-brain relationship, the biological roots of behavior, and to guide better therapies for conditions that alter cognitive and emotional health. Every image is a glimpse into the complex universe residing inside the skull, a universe we strive to comprehend with patience, rigor, and wonder. Now, as you drift towards restful sleep, let these images settle softly in your mind's eye, a vast, dynamic organ alive with electrical impulses, shimmering blood flow, and chemical conversations. Even while you rest, your brain hums gently, and somewhere in a laboratory, a scanner quietly listens, revealing a timeless dialogue between science and the essence of being human. There is a quiet wonder in contemplating the mind, that mysterious theatre where thoughts emerge and dance before fading away. Throughout human history, we have sought ways to understand this inner realm, the soft flicker of memory, the vivid colours of imagination, the silent pulses of intention. And now, in the gradual unfolding of technology, we find ourselves edging closer to an extraordinary possibility, the recording of thoughts themselves as if they were scenes on a video. To understand what this means, let us begin by recognizing what a thought truly is. At its foundation, a thought is a pattern of activity within the brain's vast network of neurons. These cells communicate through electrical signals and chemical exchanges, creating intricate patterns that correspond to everything we experience: images, sounds, emotions, decisions. The brain, in a way, is both the painter and the gallery, continuously creating internal images that sometimes mirror the outside world and sometimes reflect purely inner landscapes. Until recently, these neural patterns were like whispers behind a closed door. We could only infer thoughts indirectly through speech, actions, or the words someone writes. Even brain scans, such as functional MRI or electroencephalograms, EEGs, have been rough maps showing which areas of the brain light up during certain activities, but not the precise content of what someone is thinking. The strokes of the mind's invisible brush remained elusive. However, advances in neuroscience and technology have progressively illuminated this shadowy territory. Brain-computer interfaces, BCIs, have developed from simple tools moving cursors on a screen with thoughts, to complex systems capable of decoding imagined speech or visual imagery, albeit in a rudimentary way. Scientists have trained machine learning algorithms on neural data to reconstruct simple images subjects are viewing, or even some words they intend to say. Imagining this progression extending into the future, one can foresee a time when it might be possible to record a person's thoughts, much like a video camera records the outside world. This would not be mere translation or rough approximation, but a vibrant, continuous stream of inner experience, capturing fleeting memories, visualizations, or internal conversations as they naturally arise. The implications of such a breakthrough ripple through our understanding of self, privacy, communication, and trust. To grasp why this is both thrilling and deeply complex, consider how the brain represents information. Unlike a film camera that captures objective scenes with light and color, the brain's recordings are highly subjective. The same image can be perceived differently by two people. The vibrant red of an apple might bring joy or hunger to one person and indifference to another. A thought is imbued with the entire context of that person's history, emotions, and personality. If a device were to record thoughts accurately, it would not simply produce an external representation like a photograph. It would lay bare a raw, unfiltered experience that often is hidden even from ourselves. Many thoughts flicker by so briefly and subtly they escape conscious notice. Some moments within us are deeply private, forming the very essence of our identity. To record these would be to peer into the soul's inner chamber. This leads us naturally to the considerations of ethics and consent. How would such technology be governed to protect the sanctity of private thought? Much like the technologies that have reshaped communication, telephones, the internet, smartphones, the possibilities for misuse and surveillance could be profound. The freedom to maintain one's inner world inviolate is a cornerstone of mental privacy. When thoughts can be decoded and projected outward, new frameworks would be needed to define boundaries between personal freedom and external access. In a sense, this future challenges our most deeply held notions about what it means to be human. We tend to think of our inner life as ultimately inaccessible to others, safe against intrusion. It is a source of autonomy to hold our views, doubts, and dreams just within ourselves. Would the ability to record thoughts diminish this freedom or enhance it? On one hand, such technology could foster extraordinary empathy and understanding. Misunderstandings born of incomplete communication might diminish if we could share our precise emotions or mental imagery with others. Relationships could deepen when barriers of language or expression no longer limit us. Imagine a world where a poet's vision, not just their words, is shared directly with an audience, or where those with speech difficulties could convey complex thoughts exactly as they envision them. In medicine, the implications also tantalize the imagination. Recording and interpreting thoughts could give voice to patients unable to speak, help diagnose mental illnesses by revealing thought patterns, or assist people recovering memories lost to trauma or neurodegeneration. It might even open the door to new forms of therapy, where understanding a person's internal critic or fears becomes more direct and accessible. Yet alongside such promise, the risks loom large. Could thoughts be stolen, manipulated, or weaponized? If employers, governments, or hackers gained access to mental content, what protections could safeguard individuals? The permanence and objectivity we associate with written or recorded information could trap us in our own past, where a fleeting thought made in anger or confusion might have long-lasting consequences. This future compels us to imagine complex legal, societal, and philosophical responses, for technology will not simply evolve in isolation. It must be shaped by human values and foresight. Privacy protections would need to be robust and enforceable, ensuring that consent is explicit, data is secure, and abuses are punishable. Discussions must take place well in advance of capability, so that society's shared ethical compass guides innovation rather than lags behind it. Turning now to the scientific basis for how such recording could become reality, it centers on ongoing efforts to understand the neural code, the ways in which patterns of neuronal activity correspond to thoughts and experiences. Our brains consist of approximately 86 billion neurons, each connected to thousands of others in a dense web. The brain's language is electrical and chemical signaling, rapid pulses relayed rhythmically or sporadically across this network. Early brain-computer interfaces have used pieces of this code by capturing signals from electrodes placed either on the scalp, non-invasively, or directly on the cortex, invasive but providing cleaner signals. Using these inputs, algorithms decode intended movements or simple imagined speech. Yet interpreting complex, abstract thought is far more challenging because the signals are noisy, overlapping, and deeply context-dependent. To surmount this, researchers are employing artificial intelligence models, particularly deep learning networks inspired by the brain's own architecture. These systems can analyze patterns in vast neural datasets, discerning subtle correlations between activity and mental content. As these models improve, the fidelity of what can be decoded may rise from rough approximations to detailed mental reconstructions. We can further speculate that future devices might integrate additional sensory and physiological data, eye movement, heart rate, muscle tension, to provide richer context. They may also leverage nanotechnology, deploying microscopic sensors throughout the brain that operate with minimal intrusion, gathering otherwise inaccessible information about neuronal communication. In such a future, thoughts could be recorded continuously, producing a temporal stream, not unlike a film reel, but uniquely personal. Consider the marvel of being able to play back a vivid memory with all its sights and emotions, or reviewing an internal dialogue that once seemed intangible. The boundary between inner experience and external record could blur. Still, an important caution is in order. The brain's plasticity, the way it changes and adapts, is partly why thoughts are transient and sometimes elusive. The brain is not a tape recorder. It is more like a dynamic artist, constantly remixing and shaping perceptions and ideas. Any recorded thought would be a snapshot, filtered through the lens of that moment's biology and context. This means interpretation requires care, humility, and an acceptance of ambiguity. From a societal perspective, the prospect of recorded thoughts challenges power structures. The control over who can access such intimate data could become a new axis of inequality. Policies and frameworks emphasizing fairness, autonomy, and protection of vulnerable populations will be crucial. Education around the technology will help demystify it and empower individuals to make informed decisions. Moreover, cultural attitudes toward mental privacy may evolve. Some societies might view thought recording with enthusiasm, celebrating transparency and radical openness. Others might resist, valuing traditional notions of inner sanctity. These differing perspectives will influence how and where technologies deploy, shaping a varied global landscape of acceptance and regulation. A fascinating consequence to ponder is how recording thoughts might affect creativity and learning. If we could capture not just finished ideas, but the developmental thoughts leading to a work of art or scientific insight, we could glimpse the mind's creative process in unprecedented detail. This might revolutionize education by tailoring learning experiences based on real-time understanding of mental states. Another potential development lies in collective consciousness or sharing mental experiences beyond individual boundaries. Could groups synchronize or share aspects of their internal worlds to create new forms of collaboration, empathy, and understanding? This would reshape social dynamics, emphasizing the connectedness of human minds at a level deeper than spoken or written language. Yet it is essential to keep in mind that while the idea of recording thoughts brings fantastic possibilities, it must be approached with an awareness of complexity and respect for human dignity. The human mind is not simply an information processor. It embodies consciousness, emotion, and identity in ways that may forever transcend pure data. Future researchers and ethicists will have the delicate task of balancing curiosity and innovation with caution and wisdom. The technology of thought recording is not destined to appear automatically. It will emerge as the product of deliberate choices grounded in dialogue with society's values and needs. This journey, from understanding the brain's electrical symphony to potentially replaying the music of a single thought, reflects humanity's timeless quest to bridge the known and the unknown. As we explore ever deeper into the workings of our mind, we open doors to unforeseen vistas, new forms of connection, and profound questions about privacy, freedom, and what it means to be truly ourselves. And so, as you rest now, take comfort in the quiet marvel of your own thoughts, those invisible patterns humming in the dark, quietly weaving your world. They are yours, uniquely yours, for now. The future beckons with the promise of deeper sharing, new understanding, and fresh challenges, reminding us always of our shared humanity. May your dreams carry you softly through the boundless landscape of the mind, where every flicker of thought is a star glowing softly in the night. And when morning comes, may your curiosity, gentle and unwavering, continue its patient journey toward the mysteries still waiting to be unveiled. As our exploration of the concept of recording thoughts like celestial cinematography drifts to a close, remember to marvel at the infinity of our mind's cosmos. Each thought is a star flickering in the hush of our inner universe. Until next time, dear dreamers, keep pondering these mysteries and join me again to journey through the constellations of knowledge.